The image shown in this diagram, this picture, is of a woman receiving a PET scan. Her arm is in a lead container, lead shielded container, to protect the patient, the technician, so around here, to protect the technician from the radioactivity or release of positrons and gamma rays when the positrons meet an electron. Around the woman's head is a PET or gamma camera, special design, when the gamma rays produced when positrons meet with normal electrons, they produce two gamma rays. Those gamma rays, these two are annihilated, and out comes the gamma rays at 180 degrees. So if there's an annihilation occurs inside here, one particle will go up here, the other down here, and be detected by the computer. The computer can then check on the time it took and the amount of loss of intensity of those photons and work out how far the photon had travelled back to this point. That point's diagnosed or labelled and then sent to the computer, and that's determined. If a gamma ray hits one of these and detects using sodium iodine crystals, and there's no alternate one on the other side, then that's not counted. That could be from background radiation or some other form. So it's only when you have these coincident detections on either side that the computer then records that as part of the scan. And here you see the actual annihilation occurring in the center here. A positron released from this positron emitting substance made in a cyclotron. That positron meets an electron in the tissue of the patient nearby, very close. When they meet, they annihilate each other and produce two photons of gamma rays. If those photons are detected by the detector at 180 degrees, a signal is recorded in the computer, and they must both be coincident or occur within a certain time, and on opposite sides they must have those two gamma rays for it to be recorded. There is some attenuation, which if we remember back in year 11, attenuation is the loss of signal or loss in intensity over time. There is some attenuation, and the computer can work out the distance based on that attenuation, how far that annihilation occurred from this gamma detector and this gamma detector. Then it can locate it very accurately within the patient. And like X-rays or MRI scans, which I dealt with at the end of the topic, PET scans are painless, non-invasive, that means no surgery, and once again with surgery you risk infection and other surgical complications. So the PET scan avoids that. Unlike X-rays and MRI, they're predominantly recording structure. Unlike those, PET scans give us an idea of function of the tissues. And they do that by the uptake of materials, of our radiopharmaceuticals. For example, oxygen 15, positron emitter, fluorine 18 in as part of FTG, fluoro deoxyglucose, and that substance can be absorbed into particular organs. For example, when parts of the brain are more active, the person is reading or smelling or saying something, they'll have a much better idea of the uptake of glucose by the brain in that particular section that's being utilised. And in this diagram, this picture, you can see that happening when a person's hearing words. Part of the brain is receiving greater blood flow and absorbing more or uptaking more glucose for those cells of the brain. When they're seeing words, they'll see another part of the brain light up or hotspots because there's more of that radioisotope being absorbed by those cells or being carried there in the blood to those particular areas of the brain. You can also see here the parts of the PET process. We have production of our positron emitting substance done in a cyclotron. We have the substance labelled with the emitter, so fluorodeoxyglucose 18 for the fluoro. We have transport of that PET substance around the body. The patient is taken to a PET camera where that can be recorded. An injection is put into the person, and the PET camera then goes to work recording what happens when that substance is in the patient. Data from the PET camera is sent to a computer and forms an image like the one above here. The results are then interpreted by a diagnostician or doctor or specialist, and the data can be formed into conclusions and diagnosis. So if we were to draw this as a flow diagram, we'd have our labelled substance, so for the brain, it's labelled with a positron emitting isotope. It's introduced into the body. It then migrates to the organ that we're studying or concerned with. Positrons are emitted. The positrons are annihilated. Producing two gamma rays at 180 degrees to each other. They are detected by a gamma camera if they are coincident and at 180 degrees to each other. That information is then sent to a computer which determines the counts and the intensity of our gamma rays. The areas of the brain which produce the most gamma rays, if we're doing a brain scan here, produce the most intense or the most number of photons. And the most photons show up as an area or a hot spot on the scan. Areas with few photons, there's a cold spot, so that part of the brain is not absorbing or uptaking our positron emitter and producing the two gamma rays. That's not happening, so it's a cold spot. So when the hot spot forms, we have our scan, and then it's diagnose the conditions by a technician or doctor or specialist. This particular diagram, you can see a PET scan in the center, but around the outside you can see the gamma camera. We've got our positron annihilation here, and this distance is greater than this distance, and the computer can determine that. It also determines the coincident events, so that this one will fire off and this one will receive that gamma particle, photon, sorry, not particle, a photon is not a particle, and the computer records that. Here you can see that same thing happening here. Distance is greater than here. Attenuation is the falling off of intensity as it gets further away, this photon. And that information is sent to the computer and forms this image. It's been put in the center of this gamma camera just to give you an idea of what it looks like. A much better image of the whole thing happening. So we've got our positron emission particles, for positron radioactive isotopes or radiopharmaceuticals injected. They go round. In this case they're using a substance that's absorbed by the brain. You can see here a hotspot forms, over here again. Different areas of the brain light up depending on what the patient's doing. Here we have our annihilation and our gamma camera tubes which detect where the annihilation came from, also the distance to that annihilation. Same here and same here, so there's several different ones there being detected. Areas that are not producing a lot of gamma rays stay dark in the diagrams, so here and here. So that's basically how a PET scan image works. They use a substance that's uptaken by the organ particular. So in this case they're using FDG, and it's 18 FDG because they're using 18 fluorine as their positron emitter. The positron emission causes annihilation, producing the two gamma rays which are detected by the gamma camera. So that's basically it.